So we'll be studying the development of brain, spinal cord and pituitary gland in 4 different sessions. So this will be the first session where we'll be seeing the neural tube formation and neural crest derivatives. So these are the following topics which we are going to cover in the development of nervous system. So especially the neurulation, differentiation of neural tube, neural crest cells and their derivatives, development of spinal cord and neural tube defects, and then the functional columns of brainstem, development of medulla oblongata, pons, midbrain, cerebellum, diencephalon and cerebrum. So nervous system includes brain, spinal cord, peripheral nerves and ganglia. Whole nervous system is developed from surface ectoderm which modifies to neuroectoderm. So neuroectoderm forms the neural plate which extends from primitive to precaudal plate. So neural plate forms the neural groove and then neural folds and then the neural tube. So neural tube differentiates to give rise to the entire nervous system. You can see here this is the neural plate which is extending from primitive streak to the precaudal plate. You can see here the neural plate formation, neural groove and neural folds, and you can see at the junction of surface ectoderm and neuroectoderm there is formation of neural crystals. This is the completion of the neural tube and separation of the neural tube and neural crest. In the third week there will be thickening of central part of ectoderm. So there is formation of the neural plate and there is folding of neural plate forming the neural groove and neural folds. So there is formation of neural folds and neural crest. So fusion of neural folds to form the neural tube. So when neural tube is formed, in the cranial end there will be cranial neuropore which closes by 25th day, and in the caudal end there will be caudal neuropore which closes by 27th day. And there is complete formation of the neural tube, separation of the neural tube and neural crest from the surface ectoderm. You can see here this is the formation of the neural tube. You can see all these are somates. You can see this is the cranial neuropore and this is the caudal neuropore. So you can see the cranial neuropore and caudal neuropore. By 28th day you can see the closure of the cranial and caudal neuropores. So neural tube remains open at cranial end and at caudal end. So this facilitates circulation of amniotic fluid which provides nutrition to developing neuroectodermal cells. So cranial neuropore closes by 25th day and caudal neuropore closes by 27th to 28 days. So in the later life, the anterior neuropore or the cranial neuropore persists as lamina terminalis and the caudal neuropore persists as the terminal ventricle which lies in the caudal end of the spinal cord. The failure of closure of this neuropore results in neural tube defects. So neural tube has central cavity which forms ventricles of brain and central canal of spinal cord, and the peripheral wall forms the nervous tissue. So neural tube elongates craniocaudally. It forms 3 primary vesicles, the prosencephalon, mesencephalon and rhombencephalon. You can see here this is the prosencephalon, mesencephalon and rhombencephalon. You can see the formation of mesencephalic flexure here at the level of midbrain. And in this you can see the prosencephalon, mesencephalon and rhombencephalon, where you can see the formation of cervical flexure at the junction of rhombencephalon and the spinal cord, and you can see the mesencephalic flexure. So these 3 primary brain vesicles respectively form the forebrain, midbrain and hindbrain. So they form 5 secondary brain vesicles, namely the prosencephalon differentiates into telencephalon which gives rise to cerebral hemispheres, and diencephalon which gives rise to optic vesicle, pineal gland, thalamus and hypothalamus, and posterior part of the posterior part of the pituitary gland. The mesencephalon forms the midbrain, and the rhombencephalon is divided into cranial, metencephalon which gives rise to pons and cerebellum, and the caudal myelencephalon which gives rise to medulla oblongata. You can see here the prosencephalon dividing into diencephalon and telencephalon, the mesencephalon remaining as mesencephalon, and rhombencephalon dividing into metencephalon and myelencephalon, and you can see the appearance of the pontine flexure here. So appearance of this pontine flexure will form the rhombic lip. The rhombic lip will give rise to the cerebellum. You can see here the cerebellum being developed from the rhombic lips. So this is the mesencephalon, this is the telencephalon, this is the diencephalon, and this is the metencephalon, and this is the myelencephalon. So metencephalon gives rise to pons and cerebellum, myelencephalon gives rise to medulla oblongata. And you can see the olfactory bulb emerging from the floor of the telencephalon. So you can see here. So these are the 3 primary brain vesicles, prosencephalon, mesencephalon and rhombencephalon, and you can see the spinal cord. And these are the secondary brain vesicles. So where prosencephalon divided into telencephalon and diencephalon, mesencephalon does not divide, and rhombencephalon dividing into metencephalon and myelencephalon. You can see here lateral ventricle is the cavity of the telencephalon, the third ventricle is the cavity of diencephalon, cerebral aqueduct is the cavity of mesencephalon, and the fourth ventricle is the cavity of myelencephalon. The central canal will be the cavity of the spinal cord. So this is the further development of the brain vesicles, and this is the complete mature brain. You can see the telencephalon overgrowing the diencephalon, mesencephalon and metencephalon. So vesicles of neural tube revision: prosencephalon gives rise to telencephalon and diencephalon. Mesencephalon gives rise to midbrain, and the rhomencephalon gives rise to metencephalon, which gives rise to pons and cerebellum, and the myelencephalon, which gives rise to medulla oblongata. This is the spinal cord. Telencephalon forms the cerebral hemisphere, and diencephalon forms the optic vesicle, pineal gland, thalamus, hypothalamus, and posterior pituitary. So there are four flexures, as I've shown earlier: such cervical flexure, junction of spinal cord and hindbrain; cephalic flexure at mesencephalon; pontine flexure separating myelencephalon and metencephalon; and telencephalic flexure separating telencephalon and diencephalon. Then you can see the cavity of the neural tube gives passage to amniotic fluid till the closure of anterior posterior neuropores. So flowing amniotic fluid nourishes rapidly growing brain and spinal cord. So on the formation of the brain vesicles, cavity of neural tube gets converted into ventricles and communications. And this is as follows: like the cavity of telencephalon is the lateral ventricle, and this lateral ventricle communicates with the third ventricle through interventricular foramen of Monro. Third ventricle is the cavity of the diencephalon. Then the mesencephalon cavity is the aqueduct of Sylvius. The rhomencephalon forms the fourth ventricle. The cavity of the spinal cord forms the central canal of spinal cord and its terminal ventricle, also called as fifth ventricle or ampulla caudalis. Terminal ventricle lies in conus medullaris, the terminal part of the spinal cord. So in the roof of the fourth ventricle, there will be two foramen of Lashka and one foramen of Magendie, through which the CSF communicates into the subarachnoid space, or the ventricles communicate into the subarachnoid space. So now what happens? This is the neural crest cells, which is a very important short notes. So during invagination of the neural plate, a distinct group of ectodermal cells appear along the edges of the neural groove. So this group of cells are called neural crest cells. So in the formation of neural tube, neural crest cells come to lie in zone between the neural tube and the surface ectoderm. So neural crest cells gets divided into dorsal mass and ventral mass. You can see here the dorsal mass divides into neuroblast, spongioblasts, and pluripotent cells. Neuroblast gives rise to dorsal root ganglia, sensory ganglia of the 5th, 7th, 9th, and 10th cranial nerves, skeletal elements of the pharyngeal arches, odontoblasts of the teeth, and parapharyngeal cells of the thyroid gland. The spongioblast gives rise to satellite cells in the ganglia and Schwann cells, and pluripotent cells form the melanocytes. The ventral mass forms the sympathochromatiform organ. Sympathoblast will form the sympathetic ganglion neurons and parasympathetic ganglion neurons, especially of the ciliary, pterygopatent, submental, and aortic ganglion in the head region. The chromaffin cells will form the chromaffin cells of medulla of adrenal gland, para-aortic body, argentaffin cells in respiratory system, and enterochromaffin cells in the gut. Other derivatives of neural crest cells include the facial bones and vault of skull, dermis of the face and neck, muscles of the ciliary body, sclera and choroids of eyeball, substantia propria and posterior epithelium of cornea, pharyngeal arch cartilages, similar valves in the heart, spiral and bulbar septum in heart, and the pia mater. You can see here, this is the neural crest cells, neuroblast, bipolar neuroblast forming pseudounipolar neuron, dorsal root ganglia, and sensory ganglia of the cranial nerves. And you can see the bipolar neuron of the cranial nerve 8, and you can see the neurons of autonomic ganglia. And if you see this undifferentiated neural crest cells, they are giving rise to satellite and Schwann cells. You can see the formation of pia mater from neural crest, chromaffin cells, and the melanocytes, and the parafollicular cells, and the cartilage of the pharyngeal arches, and the odontoblasts of the teeth.